Reaction 10, pyruvate kinase. So what's going to happen here is there's going to be two ATP molecules that are basically used as the payoff of glycolysis. So up until this point, assuming everything's gone as planned, we had two priming reactions that used ATP, and then we got back two ATP molecules. So finally in pyruvate kinase, we're going to actually get two extra. This is also going to have a large negative delta G, so giving that this is going to be subject to regulation because of that. It's also going to be allosterically activated by AMP and fructose 1,6 bisphosphate. It's going to be inhibited by ATP and acetyl-CoA. As we go through, we'll also talk about the fact that this basically has two main steps in this mechanism, especially that keto-enol equilibrium of pyruvate. So looking overall at the reaction, what we have is we have phosphoenol pyruvate, which is going to then go to pyruvate, and that one phosphate is going to go on to ADP. So we are again going to have that substrate level phosphorylation. Overall, this reaction does have a very low negative delta G value, so this is going to be going in that forward direction. Here again are the our structures of this. So when it is inactive without the ligands, it's got a slightly different shape. So when it's actually bound to things like fructose 1,6 bisphosphate, it's got a slightly different shape to it. So as I mentioned, there's basically two main steps. That's why you see two sets of arrows. So the first step is getting rid of that phosphate off of phosphoenol pyruvate and onto ATP. There is a more complicated mechanism reaction for it. We're going to gloss over that here. I will talk a little slightly about it on the next slide, but for the most part, just remember that there's two main steps. The first one's getting rid of that phosphate, substrate level phosphorylation, and then the second is this enol keto tautomerization. And this tautomerization is actually the driving part. So the substrate level phosphorylation does still happen spontaneously, but just barely. That enol keto tautomerization is the part that is more favorable and helps make this entire reaction overall favorable. So the mechanism that involves the transfer from phosphoenol pyruvate to ADP does actually have 4 different steps in here. So we're not actually going to focus on this, aside from to say that, again, anytime you're dealing with a lot of phosphates and those negative charges, we're again seeing magnesium play a key role, as well as it's actually usually potassium, but there's another just positively charged metal ion that's also playing a role in helping stabilize all of the negative charges present. On to regulation. This is going to be activated by AMP. So again, it's going to be activated by a low energy molecule. It's also going to be activated by fructose 1,6 bisphosphate. So remember that that's going to be one of the products of one of the reactions from phase 1 of glycolysis. So essentially, when something's produced, it's a way of kind of telling the second phase and this other set of enzymes, hey, something's coming down the line. So it's a way of kind of saying that glycolysis is what's being activated and moving forward. Pyruvate kinase is also going to be inhibited by a high energy molecule, by ATP, and also inhibited by alanine. Alanine can actually be made from pyruvate. So again, we have that feedback, that negative feedback loop to say, hey, we have enough energy, you can stop. Hey, we have enough pyruvate, you can stop now. Liver pyruvate kinase is also going to be regulated by covalent modification. So essentially, we've now gone through all the main parts of the 2 phases of glycolysis. So as I mentioned before, but I'll say again, it helps a lot if you'll actually take the time to write out all of these steps, because there are a lot of enzymes, there are a lot of structures, there are a lot of substrates and products and things happening in here. And if you can actually put pen to paper, this can help you get a better handle on what's going on. I have this here, what's the presence of AMP indicate in a cell? Something you've probably noticed is that both AMP and ATP show up a lot in glycolysis, and they're also going to play a role when we get to gluconeogenesis. So remember that AMP is the cell's way of saying that it's low on energy. So we mentioned briefly that in the liver, pyruvate kinase is going to have a covalent modification. So that is actually done by phosphorylation. So in this case, when it is phosphorylated, it is less active. When it is dephosphorylated, then it is more active. So as this goes through, there is another protein kinase, which is activated by glucagon. So that kinase will add the phosphate. When the glucagon signal is active, that is going to make the pyruvate kinase less active. What happens when that phosphorylated enzyme happens is you have a higher Km, and it's also more easily and more strongly inhibited by ATP and alanine. So that phosphorylation actually changes the enzyme enough so that it is not going to be holding on as tightly to its substrate, to phosphoenol pyruvate, and it's going to be more easily and more strongly inhibited by ATP and alanine. So what happens to NADH and pyruvate? We made stuff, well, where does it go? What happens? Well, whenever we look at this, we have to look at both under aerobic and anaerobic conditions. So if there's oxygen available, NADH is actually going to continue onto the electron transport chain, which will make ATP an oxidative phosphorylation. So remember that we've been talking about ATP as substrate level phosphorylation up until this point. When we finally get to the electron transport chain, we'll talk about it in another way made from oxidative phosphorylation. If we do not have oxygen, if we're in anaerobic conditions, then NADH is going to be oxidized by lactic dehydrogenase. And all that's really going to do for us is it's going to just produce more NAD+. So if we have oxygen, we can actually use the NADH to get even more ATP. If we do not have oxygen, then all we're really going to do is recycle that NADH so that we can make more ATP from glycolysis. So here's a little diagram that shows that. So remember that we were using NAD+ to go from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. And so the lactate dehydrogenase is going to take pyruvate to lactate and is going to utilize that NADH that was produced. So we can get the NADH plus back to continue glycolysis. This does happen in our muscles. So whenever we're working or running really hard with our muscles, we sometimes do that then without oxygen, and our muscles will still produce energy, but if we don't get enough oxygen to them, then it'll produce it anaerobically. That's then what that high lactate will then cause cramps. So that's why you're supposed to stretch afterwards and help get some of that lactate out of your muscles. Hibernating turtles can also have this happen to them because if the turtles hibernate long enough lying in the mud, they don't get enough oxygen either, and they'll have the same thing happen. Their shells will then actually release minerals to help buffer the lactate through hibernation. Also, lactate, when it's produced, can also be produced by lactic acid bacteria in that same anaerobic condition, which will give you that acidic flavor of yogurt and sauerkraut. So that was the NADH. What about pyruvate? Pyruvate's also going to have aerobic and anaerobic options. For aerobic options, pyruvate proceeds to the tricarboxylic acid cycle, which we've mentioned before. But if there isn't oxygen, then that pyruvate's going to go to lactate, as we saw previously, or it could also be made into ethanol. Those last two, under the anaerobic conditions, are examples of fermentation, where what you're really doing is you're getting the production of ATP by reaction pathways where organic molecules function as donors and acceptors of electrons. So the other version of that is respiration. So if you have oxygen, then you use oxygen as that electron acceptor. And that is so different that that's called respiration. The other ones are versions of fermentation. Just to compare, when glucose goes to lactate by glycolysis, it has a certain value that's associated with standard delta G value of negative 122.6 kilojoules per mole. Under physiological conditions, you actually have a higher delta G value where you have about 50% of the energy released is conserved by ATP, which is pretty good for energy conservation. Notice that when you have glucose to lactate, you have no net oxidation or reduction, but you still have enough energy to sustain growth. So these pathways have been around for a while for good reason. So just looking at the reactions for glycolysis, just looking at the difference of glucose to pyruvate and glucose to lactate, you have a lot of similarities, but again, what you see is that in pyruvate, we do have that production of NADH, but when we're using lactate, that NADH gets used up. So we don't have that extra reductive power to move on with. So this is a slide that we've seen before, but again, just to remind you that there's that difference when we're looking at standard states versus the delta G, mostly based on those concentrations of products and reactants, and that as we saw, there are those low places where you have those low values of delta G are usually where you have those more regulated steps. Since we mentioned different kinds of fermentation, one of the other one in here is pyruvate going to ethanol. I like to have this on a slightly different slide just because everything else is stuff that does happen in humans. So the notion that we can have in our muscle cells, we can go to lactate. In our cells, we can also do regular respiration. Alcoholic fermentation is something that humans do not do. This does not happen in our body. We can't do this. But pyruvate can be further reduced to ethanol in yeast, which is also then a means for regenerating the NAD+. So it has the same functionality that we were using to go from pyruvate to lactate, but here instead you get both carbon dioxide produced as well as ethanol. So you can actually get some sort of mash of corn and other grains that can then be fermented by yeast, and it will produce ethanol and carbon dioxide. So this is how we do a lot of our regular alcohol production for alcoholic beverages. Other things too. As you might imagine, there are other fermentations. So there are different bacteria that can produce things like butanol and acetone. So acetone was made industrially by fermentation. So it's always kind of one of the fun things to think back throughout our history. But when we needed certain things, when either production slowed down or certain resources ran out, they actually discovered that you could get this one particular bacteria to go ahead and produce acetone. And it was really the mark of the beginning of modern biotechnology, is basically getting some sort of bacteria to scale to get a product that you would then use in industry. Getting different bacteria to produce butanol or perhaps algae has been a lot of interest as a potential fuel source. This is just to show that there's a variety of different pathways that can still undergo fermentation. So this is the Ettner-Dulldorf pathway, found in only some bacteria. Mostly what this is because you can get different places to produce ethanol, you wind up getting higher ethanol concentrations, and it does actually tolerate higher ethanol concentrations. So it's a particular interest in certain industries. It is important in tequila fermentation. So just realize that there's multiple paths that involve fermentation. Just like there's different ways for certain bacteria to process pyruvate in different sorts of fermentation, realize that you can also use other substrates other than glucose in glycolysis. There will be different things that you need in order to actually get those sugars into the pathway, which is what we're going to take a look at. So this is a good summary slide to show that fructose, mannose, galactose can all be used. Fructose and mannose are routed by fairly conventional means. Galactose, though, usually needs a little bit more help to get into the pathway. So as we're looking at this, we were just talking about fructose and mannose. So mannose just needs a few small tweaks, and it can actually enter in as fructose 6-phosphate. So after you use phosphorylate mannose to mannose 6-phosphate, it can then actually also then be changed to fructose 6-phosphate. Fructose has a couple different options, but you can actually, it has a couple options with fructokinase, and an aldolase will actually get it in at that very, very end of the first phase of glycolysis. Galactose has additional steps because it's actually going to be connected to UDP before it can actually get into the glycolytic pathway. The Lore pathway then shows that it involves additional enzymes, and especially this transfer of this UDP glucose and UDP galactose. There's that fun quote from Jurassic Park where life finds a way. So this is, to me, that life finds a way pathway. Seems to be really fairly complicated just to use galactose, but again, if that's what you have and that's what you need, there's always another way to make things work. This is just to show you what that UDP sugar looks like. Glycerol can also enter glycolysis. So glycerol is just that small 3-carbon carbohydrate, but when phosphorylated, it goes to glycerol 3-phosphate, which then can be oxidized to dihydroxyacetone phosphate. And again, it enter right at that end of phase 1 of glycolysis. So glycerol is another option. Since this is produced from the decomposition of triacylglycerols, so in the decomposition of particular fats, it's also nice that this can also enter the glycolytic pathway. So here again is the next step in that. So going from glycerol to glycerol 3-phosphate, going from glycerol 3-phosphate to dihydroxyacetone phosphate. That does involve the glycerol 3-phosphate being oxidized. So therefore we'll also produce a molecule of NADH as it goes along. Something that I'm certain that you've heard of, an important disaccharide, is lactose. So utilized by a lot of mammals, this is usually then synthesized during late-stage pregnancy and lactation. So therefore, if mothers feeding their babies are going to produce this in our placental mammals, then there's got to be a way for this to then be utilized by those infants. The synthesis of lactose, though, that's made by the mother is done by lactose synthase. The lactose breakdown then is going to be done in the intestines by lactase, which will help those newborns be provided with that galactose. Some humans are lactose intolerant due to that low level of lactase. And there is our lactase reaction. So again, breaking down our disaccharide into galactose and glucose. And that's going to be the end of glycolysis. We made it.